Please join me in welcoming Dr. Sue-Ann Ma.
So as I was growing up, personal computers and Nintendo brought video games right into our home. And this was a big deal, right? Because it truly changed how a lot of kids like me spent our free time. Now, my absolute favorite game to play was Tetris, right? The rules are so simple, but the game's so challenging. You have these shapes falling in a reservoir. You can kind of control the shapes as they fall. Your objective is to make these complete lines. So you clear them and score points. That's really simple, right? But as we all know, the problem is that we can't really control what shapes are coming into that reservoir, right? And they keep coming faster and faster as the game goes on. Eventually, they stack to the top, and it is game over, right? And so I love these games. My parents did not share my love of these games, and they were convinced that games like Tetris were gonna quote unquote rot my brain, okay? Fortunately for them, I found a love of science, and I redirected my energy towards that. I did some of my best training right here at the U of A as a PhD student. Then I went to San Francisco, and now I'm back again at the U of A as a researcher, where I'm trying to find new ways of preventing and treating Alzheimer's disease. The dream job, guys. And so in my lab, we study a protein called tau, okay? And it's one of the proteins that we think is really responsible for killing our brain cells. And a lot of you might be curious as how a single protein can actually cause so much damage. And we think what it comes down to is something about tau's shape. So lots of proteins can fold into one or two shapes, but tau has this really unusual ability to fold into dozens, if not hundreds, of different shapes. And so some of these shapes are really useful or harmless in the cell, and so we're not really worried if we have a lot of these shapes in our brain cells. But some of these shapes are actually really bad, okay? And they're bad because they stack one on top of each other to form these really rigid rod-like structures. And in our brain cells can't really get rid of them. Worse, in Alzheimer's patients, what we know is that we make more of these bad shapes and less of the good shapes. What ends up happening is you multiply these stacks, right? More and more form in your brain cells, eventually clogging them completely until they die. Now, as I thought about these deadly tau stacks, I started to see how dealing with them in Alzheimer's disease was a lot like playing a nightmare game of Tetris. So stay with me, guys. So imagine your brain cells a reservoir, and the tau protein are the pieces. And you're playing a game where the only piece you keep getting over and over again is the Z piece. The worst piece, guys. And we know it's the worst piece, right? We can see here, because it's really hard to clear the board with them, right? And so just like in the game, your cells, no matter how hard they work, the defense systems, they can't clear these tau stacks. They keep on stacking higher and higher because they're too strong, okay? Eventually, it's game over for your brain cells. But maybe some of you will remember that I said tau can take on many different shapes. Can we use that knowledge to our advantage somehow, right? Can we change the game? And what I mean by that is, like, can we make the shapes we want and less of the shapes we don't want? And if we could do something like that, right, we might give our brain cells that fighting chance to clear those tau stacks. And so this is what my lab focuses on. We try to understand the core principles of how to take that tau protein and force it to become one shape over another. So now how do we study how tau folds? Well, it's actually really hard to do in a human brain, as you guys might imagine. It's hard to do in living cells. So what we do is we take advantage of bacteria, and we turn them into protein factories to make lots of human tau protein. Then we take that protein out of the cells so we can study exactly how it folds. And the other really great thing about this bacteria is that we can also use it to change the tau protein, right? We can make genetic mutations in the tau sequence, kind of like changing the blueprint for the tau protein. So we make new tau proteins that have that change, and we can study how they fold now. Now we come to the biggest challenge in my research, which is how I actually see the shape of the tau in the test tube. How do you do it? We have some methods. They take months to figure out even a single shape. And we knew we didn't have that kind of time as a team. So what we did is we developed a faster method, which we call tau structure fingerprinting. And the way this fingerprinting method works is we add enzymes that attack the tau shape at its weak point. So what I'm talking about is, say, the loop kind of sticking out from the tau shape. And each shape has multiple weak points, and so what we end up doing is breaking that tau up into small pieces and large pieces, a set of pieces that when we collect together and we look at them and we arrange them from biggest to smallest, what we get is this pattern of tau pieces. And so since each shape will have different naturally weak points, you can imagine how you would get like a unique kind of pattern of these tau pieces for every shape. And so here I'm showing you, say you have a green shape and a purple shape, different weak points. Our method can directly tell you and confirm that they are indeed two different shapes because they produce two very different fingerprints. Okay, so I told you about the hard science now. Now I'll tell you some of the discoveries we made using this method. The first is that we know that if we put a mutation right into a specific location within tau, we can actually change its shape. So here I have a tau protein that wants to fold into a green shape. And so we try and we put a mutation circled in pink right here, and we see it doesn't really do anything to change how tau folds. It still folds into the green shape. That's sad. But don't worry, if we put the tau mutation somewhere else, right, move it to the left a little bit, now we see we can create a new tau shape. And so just by changing where we put this mutation in tau, we've been able to find ways to make over a dozen new tau shapes. And we think that some of these shapes might actually be really useful for clearing those tau stacks. Now, the other really cool discovery that I want to talk to you about today is not only can you use a mutation to change the shape of tau, you can use it to change the shape of tau proteins around it. So how do we do that? So there's two tau proteins. One makes, say, a T shape, and the one with the mutation makes a line. So what would you guys think would happen if we mix these two together, right? You might expect that you would get both T shapes and both line shapes, right? As each of the taus folds into what it's normally programmed to do. Instead, what we find is that all the shapes are line shapes, right? Even the ones that wanted to become T shapes. And this discovery is really exciting to us because it's like you've created a new Tetris piece with superpowers, right? A tau shape that can change the shape of other tau proteins just by being around them. And it's discoveries like this that really give us hope that we can find those exact right shapes that can take back control over that bad tau that's building in our brain cells. And now I'll always remember that my parents told me that video games were going to rot my brain, and if I hadn't found science, that might have happened. But I'm really glad that I played those games now, because games like Tetris might just be the source of inspiration we need to save our brain in Alzheimer's disease.